Hi guys, hope you're doing well. This is Kenna here. We're going to go ahead and continue our discussion on cell transport. So make this part two. This is active transport and osmosis. So let's go ahead and jump in and look at our essential questions. So first of all, what is osmosis? Now you may have heard this term. A friend of yours says, "Oh, I'm learning by osmosis" or something like that. But let's figure out exactly what the scientific definition is for osmosis. Number two, how is osmosis similar to and different from diffusion? We spent a lot of time talking about diffusion and facilitated diffusion last class. Let's go ahead and look at osmosis compared to that and see what their relationship is. Number three, what is the difference between passive and active transport? Now, I know we already answered this last class, but I think it's an important point and deserves to be hit on again. Okay. And number four, what other types of intercellular transport exist? All right, just like last time, I want to review this key term of homeostasis. So pause the video, see if you can come up with the definition for homeostasis. So homeostasis is the process of maintaining balance or equilibrium. Okay, and just like last time, cells have to keep the proper concentration of nutrients and water coming in and our wastes going out. This is done by four processes, two of which we covered last class. Those two were diffusion and facilitated diffusion. And today we're going to go over active transport and osmosis. So, both of the processes that we highlighted last time, diffusion and facilitated diffusion, are what we consider passive transport, meaning that there is no energy required in addition to the kinetic energy that's already in the molecules to make it happen. We don't have to add any energy. Active transport, on the other hand, does require the addition of energy on top of the natural kinetic energy. And because we need to use energy, we need to have something that's going to force this movement to occur. So we have what are called carrier proteins. These are proteins that are embedded in the membrane surface. These are intrinsic, they're going all the way through. And I want you to think about them literally grabbing the molecule on one side and pulling it to the other side. So they do change their shape as they go ahead and move the molecule from one side of the membrane to the other. Now, this process can be identified as what with the term endocytosis. Endo, that prefix means into, and cyto meaning cell, and osis process. So the process of taking into the cell. It kind of tells you what it is. So if that's endocytosis, what do you think exocytosis is? That's right, expelling something from the cell. So exo means out, like exoskeleton. Cyto still means cell, and osis still means process. So the process of exiting something from the cell. Now, when we start looking at endocytosis, we're causing something to move in. Now, sometimes we have to go ahead and do this using a little vesicle or a little surrounding bubble that's going to go ahead and bring whatever the object is into the cell. When that happens, we typically will send something like a lysosome to it to go ahead and digest it, whether it's digesting a piece of food or trying to attack and fight off an invader that it doesn't want to have inside of it. There's a particular version of endocytosis known as phagocytosis, and this is the type of endocytosis where an entire cell gets brought in. So if we have a smaller cell that's being brought inside a larger cell, this is where that term comes from. So receptor mediated endocytosis occurs when we transport material that's bound to the surface and pull it into it. And so when we start thinking about the transport of insulin, we start thinking about cholesterol coming into animal cells. These are examples of this process. In exocytosis, material is collected into a vesicle and then shipped out of the cell. This is very similar to what the Golgi body or Golgi apparatus does when it's packaging or shipping our proteins outside of the cell. So let's take a look at this. So in this particular instance, in the upper right-hand corner, what you're looking at is food particle that's being taken in by endocytosis. So here you go, there's your food particle. Notice that it gets bubbled in terms of a vesicle. Now it's in the vesicle that fuses with a lysosome, has all these digestive enzymes in it. Remember, enzymes are proteins. It breaks down the food particles. It releases the products of digestion, so the food particles that it wants to keep get released into the cell, and the ones it doesn't need get pushed back out, and this little vesicle rejoins with the plasma membrane, pushing the waste out into the environment around the cell. Okay. Now let's go ahead and move on to osmosis. Now, if diffusion is the movement of materials from an area of high concentration to an area of low concentration, and the same is true for facilitated diffusion, active transport can really go wherever it wants because it's using energy. So there's no real limits to what active transport can do. It's typically actually going to go from a concentration of low to high because otherwise the body would likely use diffusion because it doesn't need to use energy for it. So the last one we're going to really focus on today is osmosis. Now, osmosis is the diffusion of water across a selectively permeable membrane. Okay. And this is going to move until the water is balanced on both sides. And so if we go back to a little paint version of a membrane like we did from last time, okay, you'll notice that we have a low concentration in this particular example of water molecules on one side and a high concentration of water molecules on the other side. Now, we should know from our discussion last class that small things like oxygen, carbon dioxide, and water can always go through the membrane. Okay, they can squeeze in between the phospholipids and do not need the assistance of some sort of carrier protein like an active transport or transport protein like we typically would see those tunnel proteins for facilitated diffusion. So over time, the water molecules are going to move across in this case because you'll notice the larger molecules, say sugar, are not able to fit through the membrane itself. And so it's going to continue to move water molecules across. And you'll notice that the point where it gets comfortable, where it identifies itself as being at equilibrium, is not when the number of particles is the same on both sides, but instead it tries to move them across until the concentration or percentage of water to sugar is the same on both sides of the membrane. Because when you start to think about it, one cell versus the rest of your body, we want to kind of spread out that material, not have it all go into a single cell or pull it all out of a single cell. And so your body primarily functions based on these concentration gradients, percentage wise, rather than doing raw numbers. So let's review those terms that we gave you last class going over cell concentrations. Let's start out with hypertonic. Do you guys remember what hypertonic means? Good. So hyper means above or beyond. So hypertonic means more dissolved solute. Okay. So what about hypotonic? Good. So if hyper is more, hypo must mean less dissolved solute. And then we have isotonic. What about iso? If you remembered your geometry, think about isosceles triangles. Iso means the same dissolved solute. Okay, so some key things here that if you're not remembering these terms, we make sure that we go ahead and get them under our belts. Okay, when we go ahead and look at this, we've got this term solute. Solute is what's being dissolved. This is why this ends up being so important. So this is what's being dissolved. Okay, dissolved. And the solute is going to be dissolved into the solution. Excuse me, the solvent. Okay, so solute is dissolved, solvent does the dissolving, and together those produce a solution. Let's go ahead and do our drawing tool. So solvent plus solute come together, and that's going to produce a solution. So this is where we get a little trickier with osmosis. We have to remember that in living systems, okay, in living systems, water, water is the typical solute, solvent. Water is the typical solvent. Okay. So when we start looking at these terms hypertonic and hypotonic, you'll notice that all of them deal with solute, 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 not with solvent. Okay. Which water is typically the solvent. So what was dissolved in it is going to be your sugars, your sodium ions, your whatever it has to be. Okay. So water is the typical solvent. Now, what we have here is a freeze fracture of an actual cell membrane. I've been telling you guys all along just how incredible proteins are and all the cool things that they do. Well, hopefully, you realize just how much of a role these proteins play inside the membrane in terms of transport. So what you see here is they've actually taken a freeze fracture of a cell membrane. So they dipped the cell essentially into liquid nitrogen and then they cracked it. And when it cracked, you can actually see under the electron microscope the two different surfaces. You've got one inner layer and one outer layer of the actual membrane. And all those little bumps, those little circles that you kind of see in there, those are each their different types of proteins. And so you still can't see the phospholipids themselves. They're just too small. Okay, but you can see very clearly here that there are two layers, that lipid bilayer we talked about, and you can see that there are a ton of these embedded proteins to allow for the transport of specific materials. All right, how are we going to finish off today? So today we want to go ahead and compare the four different processes we've introduced you to. Number one, diffusion. That's the one we introduced you to first, last class. This is a passive form of diffusion. Do you guys remember what passive means? Okay, so passive is going to mean no energy required versus active, which is going to require energy. Okay, so diffusion and facilitated diffusion should be no surprise that they're passive. They just rely on the energy that's there. And since this is diffusion, still says diffusion, it's going to be passive. Osmosis, if you remember our definition, is also a form of diffusion, but it's the diffusion of water. And that's where it gets a little tricky because it's moving water, but the terms hypertonic, hypotonic, and isotonic are with respect to the solutes, not the solvent. The last one on here is active transport, and it's the only one that actually uses energy. And so you'll notice if you can answer these correctly, you should also be able to answer these correctly because they are directly related to each other. Okay, so if we're dealing with this idea of active and passive, okay, active and passive right here, this is the same answers as is energy needed because that's the definition, remember, of passive and active. Passive means no energy is required. Active means it requires energy. Then we go ahead and start looking at what type of molecules actually move through. With simple diffusion that doesn't need the help of a protein, you're looking at small things, typically hydrophobic or nonpolar. This is why we're looking at things like oxygen, carbon dioxide, water. They have to be really small. And they're always going to go ahead and go down the concentration gradient. Okay, down the concentration gradient, remember, is high to low. I'll put C for concentration. Okay, again, no energy is needed, that we defined with it being passive. And then remember that diffusion just goes between the phospholipids itself. It does not need a protein to help it get across. Okay, so no protein is needed. Osmosis is the diffusion of water. And so it really is the same on almost every front. The difference is, excuse me, the difference is that it's only for water. And because water is typically the solvent, not the solute, it goes towards the higher concentrations of solutes. So this big job is really going to dilute solutes. All right. So in some respects, you can also think of this one as going from low to high, but it's low to high with respect to the concentration of the solute. It still goes from high to low with respect to water. Okay? Because remember, it is diffusion. So osmosis still goes from high to low with respect to water, but goes from low to high with respect to the solutes. And again, water is a tiny molecule, so it can actually go between the phospholipids. It does not need the assistance of a protein. Then we get to facilitated diffusion. Now, it is straight up diffusion, so just like diffusion, it's going to go from high to low concentration. Okay, so high to low concentration. And what kind of molecules are this? Well, each type of facilitated diffusion protein is tailored for a specific type of molecule. Okay, so it does require a protein. So we want to go ahead and highlight this here. But we do require a protein. Okay, different types of proteins. Here we're dealing with a simple tunnel versus a carrier protein, which is going to go ahead and grab it when it comes to active transport. Okay, so when we start looking at this, we have to make sure that we're really paying attention to what we see here in our diagram. Facilitated diffusion needs a protein because the molecule is either too big or too charged to go ahead and fit through, but it doesn't need any extra energy because remember, it's still falling through. Okay. When we get to active transport, however, we need that energy. And so this is almost always going to be from low to high. It doesn't have to be, it can really be whatever it wants, but it's typically going to be from low to high concentration. And that's why the body's willing to go ahead and expend energy to make it happen. Okay. So notice here does require energy, and that matches up with this idea of it being active. All right, guys, so that's all I've got for you today. I hope you're doing well. Have a great rest of your day, and I'll talk to you soon. Take care. Bye bye.